Welcome students, I am Dr. Gandhi and today we are going to study photosynthesis. I am going to teach you photosynthesis in two parts. The first part then will be experiments which are required to be studied for photosynthesis, and the second part will include the process of photosynthesis itself. Now why have I taken first the experiments on photosynthesis? That is because there are two conceptual words. If you understand those two words, then the experiments are very, very easy. You don't require to study. In fact, if you make an attempt to genuinely understand what I'm trying to say by meaning of these two words, you don't require to study till your boards and maybe even further. The two conceptual words are de-starching and starch test. I repeat, the two words on which today's experiments will be based are de-starching and starch test. So with this bit of information that today we are going to study experiments based on photosynthesis with the words clearly understood, de-starching and starch test, you will realize how easy the experiments become and obviously interesting also. With that, I take you to the board and begin with the basic requirement of photosynthesis, the definition and the equation. So please follow me on to the board. So what is the definition of photosynthesis? Photo means light and synthesis means to join. So in the presence of light, two substances will be joined, carbon dioxide and water. And what will be synthesized? Glucose. Okay? For that, the requirements are light and chlorophyll. Photo means light. So I have written the equation and I have decided to teach you the definition of photosynthesis in 5 parts so that it becomes easy. So when you count them on your fingers, because this is one which comes in the exam along with the definitions and the equation. You are supposed to write and define and the balance. So therefore equation has to be balanced. Therefore I have made an attempt to divide it into 5 pronged, we call it. Okay, so we begin. Photosynthesis then is a process occurring in all living cells containing chlorophyll. A process occurring in all living cells containing chlorophyll, whereby food substances are produced, glucose, starch, using carbon dioxide and water, energy of light, oxygen is released as a byproduct. Simple. So we begin again. It is a process occurring in all living cells containing chlorophyll. Second, whereby food substances, glucose, starch are produced, using carbon dioxide and water, energy of light, oxygen is released as a byproduct. This then is the balanced equation. Please remember, you must include light and chlorophyll on the arrow. Okay. Now I told you the two words required for the experiments on photosynthesis. The external factors. What are the external factors? Light, chlorophyll, carbon dioxide. There are 3 experiments which you are supposed to study. Light is required for photosynthesis. Chlorophyll is required for photosynthesis. Carbon dioxide is required for photosynthesis. The factors which are required, pay attention, for photosynthesis. And you are going to now understand the word de-starching and starch test. We begin with the first experiment. But before that, I will have to give you a little detailed idea about how starch is identified. Okay, so I will now erase this. So now, carbon dioxide, pay attention, carbon dioxide and water. I'm not going to balance because I just want you to know the factors required for photosynthesis. Light energy, chlorophyll. What is going to be produced? What is going to be produced? Glucose. And what is going to be released? Oxygen. Pay attention. As soon as glucose is produced, it is stored as starch. When I take personal classes, once the school reopens, I'll make you understand why glucose is stored as starch. Okay. What is the test to identify the presence of starch? You have to use a solution. The solution used to identify starch is called iodine solution. So to an unknown substance or on a leaf in our case, if you add iodine solution, so I'll write down here, to a leaf, if you add iodine solution, if you get a blue color, that indicates starch present. And to a leaf, when you add iodine solution, you get a brown stain. You get a reddish brown stain. Brown stain. That indicates that the starch is absent. So now we will begin with our experiments. So now I'm going to set up an experiment. The first one is to demonstrate light is required. At the end of the experiment, we will add iodine solution to the leaf. If the part of the leaf turns blue-black, that part has starch, indicating thereby that whatever was the factor was required for photosynthesis. And when adding iodine solution, if you get brown stain, it means starch is absent. Okay, so the solution then used is a iodine solution. So now I'm going to set up an experiment. Pay attention. To demonstrate that light is required. So what I did was I took a potted plant. This then is the leaf. A leaf. Now I want to demonstrate that light is necessary for photosynthesis. Light is required. So what did I do? On this I put a black paper. So I covered the surface of the leaf with black paper. Both surfaces of the leaf, pay attention. Both surfaces, I will show it to you. Both surfaces of the leaf with black paper. So you can see one surface, the other side you cannot see. So now, why did I do this? To indicate that if I demark this as area X and this area as Y, then in the area Y, what is not being received? Light is not being received. So when I conduct the iodine test for starch, this area will turn blue. That is what I have presumed. So I am now conducting experiment 1 to demonstrate light is required for photosynthesis. What I did was, before exposing to the sun, I covered the two surfaces. So you can see now that I have covered both surfaces of leaf with a black paper. I will show you. For example, if this is one surface, I have covered it with black paper. I have also covered the other surface with black paper. So there is black paper on both surfaces. I then expose it to sunlight. So now I expose the plant to sunlight. So obviously you can see the sunlight does not reach the area Y because of black paper. It is only available to area X. So after the experiment, when I break the leaf, and demark the area as X, X and Y, and I add iodine solution, my knowledge about photosynthesis says that where there is light, there will be starch. So there will be starch in the exposed areas and there will be no starch in the covered area for a simple reason that I had covered with black paper and light was not available. This is what I had thought. But the problem is the moment I added iodine solution, pay attention, the whole leaf turned blue on adding iodine solution. That means light is not required for photosynthesis because although the paper had covered area Y, why should it turn blue? I have to understand something. There is something wrong in my understanding of the experiment. So I will now make you understand what was the mistake that we have been doing. So pay attention. I'm taking you back to the leaf. This was the leaf which I had wanted, pay attention, to demonstrate light is required for photosynthesis. But I did not realize that already, pay attention, starch is present in the whole leaf. So even if I cover with a black paper, it will still turn blue because starch is already present. So therefore, before conducting an experiment to demonstrate the requirements, light, chlorophyll, and carbon dioxide, you will have to make sure that starch is not present. And that is what I have told you in the beginning, that word known as de-starching the plant. So pay attention, now going back to it. So now I'm coining the word de-starching the plant. So, what happened in the beginning when we did first time? We did not realize that the starch is already present in all parts of the leaf. So even if I cover a certain part with black paper, it will still turn blue on addition of iodine. So now, prior to conducting an experiment, I have to de-starch the plant. What does it mean? I have to take all the existing starch away and move it to, in this case, the root, for example, which is the storage area. So I have to remove all the starch present in the leaf to the storage areas, so that before I begin the experiment, I have to ensure, remember, I have to ensure that the experimental leaf is, and I'm writing it in a different color so that it remains with you, free of starch. So prior to conducting the experiment on photosynthesis to demonstrate the presence of light, chlorophyll or carbon dioxide required, I have to take a step known as de-starching the plant. What does it mean? Removal of all pre-existing starch to storage area. So in this case, I'm going to make a visual. So I'm going to move all the starch present, removal of all starch from the leaf. Correction, from the leaf to storage areas. So I'm going back to the leaf so that you understand. So you can see there is all pre-existing starch in the leaf. I have to de-starch the plant, remove all the starch and take it to the storage area. How is it to be done? So there is a method. This is the meaning. I am now telling you what is the method. The method is very simple. Place the whole plant in a dark place. It can be a cupboard, it can be a dark room for 24 to 48 hours. This then is the method, this is the meaning, and this is the reason. So we begin again. First, before conducting any experiment to demonstrate the requirement of external factors like chlorophyll and carbon dioxide for photosynthesis, before setting up the experiment, we have to de-starch the plant. What does it mean? Removal of all pre-existing starch from the leaf to the storage areas. So now how does it happen? I take the whole plant and place it in a dark place for 24 to 48 hours. By that, what have I ensured? That the experimental leaf is free of starch. So now I come back here. So now, before covering, so pay attention. Now, with this basic idea, I'm now coming back to the experiment. So you can see now that this then is a leaf on a plant which has been de-starched. What does it mean? There is no starch on the leaf. Now I cover it with a black paper. So now I have covered it with a black paper. So obviously there is no starch in the leaf. There is no starch in the leaf. I now mark the area X, Y and X. Now I create sunlight. So this is the time when I create now sunlight. So the first step is to de-starch the plant. Second step in the experiment, expose to sunlight for a few hours, 4 hours. Now you can see the light reaches. Where does the light reach? The light reaches area X. Where did it not reach? The Y. So now at the end of the experiment, pay attention. I now break the leaf. I now break the leaf and mark the areas. Okay. And when I mark the area, I already know this area is X. This area is X and this is Y. I now add to this, I add iodine solution. So when I add iodine solution, obviously I expect that here there is starch formed in the presence of light. In the area marked X there is starch and here there is no starch. So with a prejudiced idea, when I added iodine, I expected that these two will turn blue-black and this area will turn brown, confirming that light is required. Okay? But the problem is, when I did this experiment, I did not get blue color. The leaf stained brown. Now what happened? So that means something else is missing. Apart, I de-starched, I exposed to sunlight, and I know that the starch has been formed here. Then why is it not getting a blue-black color? So now I require to create another understanding. So what is that other understanding? So I'm now creating another understanding. So pay attention. Having de-starched the plant, okay, to make sure that there is no starch, I exposed it to the light. Fine. When I exposed it to the light, the exposed areas did contain starch. So when I added iodine, I expected that to turn blue-black. Then why did it not turn blue-black? Why did it not turn? That is because photosynthesis occurs in all living cells, in a living cell. So now I'm drawing this. Look here, the same thing I'm drawing here to give you an idea. So look here, the same leaf. This is that area. This is X. This is X. This is Y. I'm drawing a cell here. This is the cell made up of cell wall. This then is the cell membrane. Now, when I added, pay attention, when I added iodine solution, and pay attention, there is starch here, pay attention, starch has been formed, okay? You can see the starch has been formed. Where has it been formed? In a cell. Next, when I added iodine solution, why did it not turn blue? So what is iodine solution? Iodine dissolved in water. So when I added iodine solution marked as iodine and water, the cell wall allowed the iodine and the water to pass, because cell wall is freely permeable, but the cell membrane only allowed water to pass. Water does not give color. The iodine remained outside because cell membrane only allows solvent. That is the reason why on adding iodine solution I did not get a blue-black color. I will repeat everything again. When I was told that I'm supposed to de-starch a plant to make sure that the experimental leaf is free of starch, I de-starched the plant. Then I covered it with a black paper to demonstrate that light is required for photosynthesis. Then I marked the areas X, X and Y. I exposed the plant to sunlight for 4 hours. And after that I was very sure that when I add iodine solution, this part where starch has been formed, this part where starch has been formed will definitely turn blue, which did not happen. So why did it not turn blue? So this is the explanation. The starch is produced in a living cell. What is the cell made up of? Cell wall and cell membrane. What is iodine solution? Iodine dissolved in water. So pay attention, when you added iodine to the cell, remember the iodine and the water both pass through the cell wall because it is freely permeable. After that, only water entered. What is the reason? Iodine is a solute and semi-permeable membrane does not allow the solute. So, if you want your experiment to be correct, you will have to destroy the semi-permeable membrane. Pay attention again. After you have exposed to sunlight and before you add iodine, you will have to destroy. Cell membrane. Otherwise, the iodine will not reach. And this is done by means of a test. So now, at the end of exposure to sunlight, to destroy the cell membrane, you carry out a test called starch test. So now we will understand what is starch test. So the 3 requirements then for my experiment to be successful, pay attention, for my experiment to be successful, there are 3 requirements. First, I should de-starch the plant to ensure that the experimental leaf is free of starch. Next, I must expose the plant to sunlight. Obviously, photosynthesis for a few hours. And then carry out the starch test, which is aimed at destroying the cell membrane, making the cell permeable to iodine. The starch test involves 4 steps. But now I'm going to teach you the 4 steps of starch test. Okay, I'm going to keep this experiment as it is, so that when we come back, having understood the 2 conceptual words of today's class, de-starching and starch test, see how easy the experiments become. So now I am conducting what is known as starch test. It is conducted in 4 steps. It is conducted in 4 steps. First, break the leaf. So I broke the leaf. Break the leaf. After having exposed to sunlight, break the leaf. And this is the main reason for the starch test. Dip in boiling water. You have to dip it in boiling water. Why did I dip it in boiling water? To destroy, to kill the cells. And when the cells are killed, iodine can easily enter the cell. So I had to kill the cell to destroy the semi-permeable membrane so that iodine can enter the cell. And if at all there is starch, it will give me a test for all the starch tests. Next, so the first key step then is to break the leaf and dip in boiling water to kill the cells so that when I add iodine, the iodine should reach the cell, which I have told you, by destroying the semi-permeable membrane. In step 2, I take this leaf, pay attention, I take the green leaf, obviously green parts of a plant produce photosynthesis, and dip it in a test tube. This then is a test tube containing methyl alcohol. I have to place it in a water bath. I place the test tube in a water bath so that when I put a Bunsen burner, I am heating the methyl alcohol is indirectly heated. Because if you directly heat it, it will explode. Remember. Next, I take this green leaf which was dipped in boiling water and place it here. In the second step, what is the function of methyl alcohol? It dissolves chlorophyll. So now the chlorophyll is dissolved. So now you can see the leaf. By dissolving chlorophyll in methyl alcohol, you have a white leaf. But the problem is, why do you want the white leaf? So that when I add iodine solution, I should be able to see the blue, black or brown color easily. On a green color, it is not possible. On a white color, things are easy to visualize. Next, step 3. Now I have a white leaf, but the problem is when you dip it in methyl alcohol, the leaf becomes brittle. What does the word brittle mean? Brittle means which shatters easily, like crystal. So if I have a leaf and if I add iodine solution which has been dipped in methyl alcohol, the leaf will shatter. I will not be able to see the color change, neither will I be able to conduct my starch test. Therefore, I re-dip in water. The third step, re-dip in water. So now the white leaf becomes white and soft leaf. So this then is the third step. And in the end, that is step 4 now. So now step 4. Now it is very easy. To this white leaf, what do I add now? Iodine solution. And obviously what will I get? Those areas containing starch will turn blue-black. And those areas which do not contain starch on the leaf or in the cells of the leaf will turn brown. So that is the 2 words which I have been telling you from the beginning of the class. The meaning of the word de-starching and starch test required for experiments to demonstrate the requirement of external factors for photosynthesis. So I come back here. Again I begin. So first I have to de-starch the plant. What is the meaning? Removal of all existing starch from the leaf to the storage areas by placing the plant in a dark place for 24 to 48 hours, thus ensuring that the experimental leaf is free of starch. Next, I set up whatever experimental need is required. For example, in this case, I set up black paper on both surfaces. Now that I was sure that the leaf does not contain starch, I now covered it with black paper, okay, and exposed to sunlight for 4 hours. So the sunlight reached the areas X, did not reach the area Y. And in the end, what did I conduct? A starch test to make sure that the iodine solution, when poured on the leaf, the iodine reaches the cell. By conducting a starch test, the main step being break the leaf and dip in boiling water. The boiling water kills the cells, destroys the semi-permeable membrane, and makes the cell permeable to iodine. Next, I took the leaf and dipped in indirectly heated methyl alcohol. What does indirectly heated methyl alcohol dissolve? Chlorophyll. What did the leaf turn? White. Next, the white leaf had turned brittle because of dipping it in methyl alcohol. So now what did I do? I re-dipped in water, so the leaf became soft. And finally you had iodine solution. If starch is present, you will get a blue color, and if starch is absent, you will get a brown color. So the requirements then are de-starching and starch testing. Okay, so now that we have got a clear idea about de-starching and starch testing, we are now going to take up a challenge. What is the challenge? I'm going to finish the experiments within 3 minutes. So when you also read, after having understood, see whether you can rehearse and write or explain those experiments in 3 minutes. If that is the case, you don't require to study till your exam. Any exam on photosynthesis. We now begin the study of photosynthesis, experiments required for photosynthesis. The experiments then are to demonstrate light, chlorophyll, and carbon dioxide required for photosynthesis. You can now see that I'm putting up a chart, and now you can see. So I put up a chart now. First, what am I trying to demonstrate? Light is necessary. What did I first take? A de-starch plant. Next, what did I expose it to? Sunlight. I hope you understand. Next, what did I conduct the starch test? In the exam, they will not ask you to write. It is understood that you know it. What will be your result? Obviously, the exposed areas on adding iodine solution, the exposed areas will turn blue and the covered area will turn brown, thus demonstrating that light is required for photosynthesis. The key words then being de-starching and starch test. Second experiment. I have taken a leaf with two areas. You can see here a green area and a non-green area. Such a leaf is called variegated leaf. Example of a variegated leaf is a plant called croton. Now, what is this experiment to demonstrate? Chlorophyll is required. So first, what did I do? De-starch the plant. Then what did I expose it to? Sunlight. What did I carry out the starch test? And I noticed the green areas turn blue, the non-green areas turn brown, confirming that chlorophyll is required for photosynthesis. And the last experiment is to demonstrate carbon dioxide is required. So now, look here. I have de-starched the plant. Before exposing to sunlight, I have covered half leaf in a beaker containing potassium hydroxide. What is the function? It absorbs carbon dioxide. So you can see inside the leaf, the part of the leaf inside the flask has no carbon dioxide. Next, after the de-starching plant and setting up, I am now exposed to sunlight and carry out the starch test. Obviously, you will now realize that the part of the leaf outside turns blue-black. The part inside will remain brown. That is because carbon dioxide is not there. Carbon dioxide is required for photosynthesis. So you can see now how easy the experiments have become. Okay? So that takes care of the understanding of the word de-starching and starch test. The last experiment is to demonstrate oxygen is released in photosynthesis. Now this is very, very easy. You take a beaker. On the beaker, you take a funnel, a short stemmed funnel, and put a water plant hydrilla. On that, invert a test tube containing water. Expose it to sunlight. In the presence of sunlight, photosynthesis occurs, releases oxygen in the form of bubbles occupied by a gas. So, how do you know that this gas is oxygen? Take out the test tube, put a glowing splinter. If it rekindles, you have confirmed that the gas is oxygen released by a process called photosynthesis. So this then is the first part of my lecture on photosynthesis based on experiments and two words. I repeat again, de-starching and starch test. Stay home, stay safe, stay blessed.